that is, from RNA to DNA. Can you suggest a simple name to the process? 6.1.2 Packaging of DNA helix taking the distance between two consecutive base pairs is 0.34 nanometers, 0.34 times 10 to 9 m. If the length of DNA double helix in a typical mammalian cell is calculated simply by multiplying the total number of BP with distance between two consecutive BP, that is, 6.6 times 109 BP times 0.34 times 10 to 9 mpb, it comes out to be approximately 2.2 meters. A length that is far greater than the dimension of a typical nucleus, approximately 10 to 6 m. How is such a long polymer package in a cell? If the length of E. coli DNA is 1.36 millimeters, can you calculate the number of base pairs in E. coli? In prokaryotes, such as E. coli, though they do not have a defined nucleus, the DNA is not scattered throughout the cell. DNA being negatively charged is held with some proteins that have positive charges in a region termed as nucleoid. The DNA in nucleoid is organized in large loops held by proteins. In eukaryotes, this organization is much more complex. There is a set of positively charged, basic proteins called histones. A protein acquires charge depending upon the abundance of amino acids residues with charged side chains. Histones are rich in the basic amino acid residues lysine and arginine. Both the amino acid residues carry positive charges in their side chains. Histones are organized to form a unit of 8 molecules called histone octamer. The negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called nucleosome, figure 6.4a. A typical nucleosome contains 200 BP of DNA helix. Nucleosomes constitute the repeating unit of a structure in nucleus called chromatin, thread-like stained pillared body seen in nucleus. The nucleosomes in chromatin are seen as beads on string, structure when viewed under electron microscope, M figure 6.4b. Theoretically, how many such beads nucleosomes do you imagine are present in a mammalian cell? The beads on string structure in chromatin is packaged to form chromatin fibers that are further coiled and condensed at metaphase stage of cell division to form chromosomes. The packaging of chromatin at higher level requires additional set of proteins that collectively are referred to as 2022-23. 100 biology non-histone chromosomal NHC proteins. In a typical nucleus, some region of chromatin are loosely packed and stains light and are referred to as euchromatin. The chromatin that is more densely packed and stains dark are called as heterochromatin. Euchromatin is said to be transcriptionally active chromatin, whereas heterochromatin is inactive. 6.2 The search for genetic material. Even though the discovery of nuclein by Mature and the proposition for principles of inheritance by Mendel were almost at the same time, but that the DNA acts as a genetic material took long to be discovered and proven. By 1926, the quest to determine the mechanism for genetic inheritance had reached the molecular level. Previous discoveries by Gregor Mendel, Walter Sutton, Thomas Hunt Morgan and numerous other scientists had narrowed the search to the chromosomes located in the nucleus of most cells. But the question of what molecule was actually the genetic material had not been answered. Transforming principle. In 1928, Frederick Griffith, in a series of experiments with Streptococcus pneumoniae, bacterium responsible for pneumonia, witnessed a miraculous transformation in the bacteria. During the course of his experiment, a living organism, bacteria, had changed in physical form. When Streptococcus pneumoniae pneumococcus bacteria are grown on a culture plate, some produce smooth shiny colonies S while others produce rough colonies R. This is because the S strain bacteria have a mucus polysaccharide coat, while R strain does not. Mice infected with the S strain virulent die from pneumonia infection, but mice infected with the R strain do not develop pneumonia. Griffith was able to kill bacteria by heating them. He observed that heat-killed S strain bacteria injected into mice did not kill them. 1E2022-23. 101 molecular basis of inheritance injected a mixture of heat-killed S and live R bacteria, the mice died. Moreover, he recovered living S bacteria from the dead mice. He concluded that the R strain bacteria had somehow been transformed by the heat-killed S strain bacteria. Some transforming principle, transferred from the heat-killed S strain, had enabled the R strain to synthesize a smooth polysaccharide coat and become virulent. This must be due to the transfer of the genetic material. However, the biochemical nature of genetic material was not defined from his experiments. Biochemical characterization of transforming principle. Prior to the work of Oswald Avery, Colin MacLeod and Maudslin McCarty, 1933-44, the genetic material was thought to be a protein. They worked to determine the biochemical nature of transforming principle in Griffith's experiment. They purified biochemicals, proteins, DNA, RNA, etc. from the heat-killed S cells to see which ones could transform live R cells into S cells. They discovered that DNA alone from S bacteria caused R bacteria to become transformed. They also discovered that protein-digesting enzymes, proteases, and RNA-digesting enzymes, RNases, did not affect transformation, so the transforming substance was not a protein or RNA. Digestion with DNAs did inhibit transformation, suggesting that the DNA caused the transformation. They concluded that DNA is the hereditary material, but not all biologists were convinced. Can you think of any difference between DNAs and DNAs? 6.2.1 The genetic material is DNA. The unequivocal proof that DNA is the genetic material came from the experiments of Alfred Hershey and Martha Chase, 1952. They worked with viruses that infect bacteria called bacteriophages. The bacteriophage attaches to the bacteria and its genetic material then enters the bacterial cell. The bacterial cell treats the viral genetic material as if it was its own and subsequently manufactures more virus particles. Hershey and Chase worked to discover whether it was protein or DNA from the viruses that entered the bacteria. They grew some viruses on a medium that contained radioactive phosphorus and some others on medium that contained radioactive sulfur. Viruses grown in the presence of radioactive phosphorus contained radioactive DNA but not radioactive protein because DNA contains phosphorus but protein does not. Similarly, viruses grown on radioactive sulfur contain radioactive protein but not radioactive DNA because DNA does not contain sulfur. 2022-23. 102 biology radioactive phages were allowed to attach to E. coli bacteria. Then, as the infection proceeded, the viral coats were removed from the bacteria by agitating them in a blender. The virus particles were separated from the bacteria by spinning them in a centrifuge. Bacteria which was infected with viruses that had radioactive DNA were radioactive, indicating that DNA was the material that passed from the virus to the bacteria. Bacteria that were infected with viruses that had radioactive proteins were not radioactive. This indicates that proteins did not enter the bacteria from the viruses. DNA is therefore the genetic material that is passed from virus to bacteria. Figure 6.5. Figure 6.5 The Hershey-Chase Experiment 6.2.2 Properties of Genetic Material DNA versus RNA. From the foregoing discussion, it is clear that the debate between proteins versus DNA as the genetic material was unequivocally resolved from Hershey-Chase experiment. It became an established fact that it is DNA that acts as genetic material. However, it subsequently became clear that 2022-23. 103 Molecular basis of inheritance in some viruses. RNA is the genetic material, for example, tobacco mosaic viruses, QB bacteriophage, etc. Answer to some of the questions such as, why DNA is the predominant genetic material, whereas RNA performs dynamic functions of messenger and adapter has to be found from the differences between chemical structures of the two nucleic acid molecules. Can you recall the two chemical differences between DNA and RNA? A molecule that can act as a genetic material must fulfill the following criteria. I. It should be able to generate its replicate replication. E. It should be stable chemically and structurally. E. It should provide the scope for slow changes mutation that are required for evolution. IV. It should be able to express itself in the form of Mendelian characters. If one examines each requirement one by one, because of rule of base pairing and complementarity, both the nucleic acids DNA and RNA have the ability to direct their duplications. The other molecules in the living system, such as proteins, fail to fulfill first criteria itself. The genetic material should be stable enough not to change with different stages of life cycle, age, or with change in physiology of the organism. Stability is one of the properties of genetic material was very evident in Griffith's transforming principle, itself that heat, which killed the bacteria, at least did not destroy some of the properties of genetic material. This now can easily be explained in light of the DNA that the two strands being complementary if separated by heating come together when appropriate conditions are provided. Further, 2-pheto group present at every nucleotide in RNA is a reactive group and makes RNA labile and easily degradable. RNA is also now known to be catalytic, hence reactive. Therefore, DNA chemically is less reactive and structurally more stable when compared to RNA. Therefore, among the two nucleic acids, the DNA is a better genetic material. In fact, the presence of thymine at the place of uracil also confers additional stability to DNA. Detailed discussion about this requires understanding of the process of repair in DNA, and you will study these processes in higher classes. Both DNA and RNA are able to mutate. In fact, RNA being unstable, mutate at a faster rate. Consequently, viruses having RNA genome and having shorter lifespan mutate and evolve faster. RNA can directly code for the synthesis of proteins, hence can easily express the characters. DNA, however, is dependent on RNA for synthesis of proteins. The protein synthesizing machinery has evolved around RNA. The above discussion indicate that both RNA and DNA can function as 2022-23. 104 Biology genetic material, but DNA being more stable is preferred for storage of genetic information. For the transmission of genetic information, RNA is better. 6.3 RNA world. From foregoing discussion, an immediate question becomes evident: which is the first genetic material? It shall be discussed in detail in the chapter on chemical evolution, but briefly, we shall highlight some of the facts and points. RNA was the first genetic material. There is now enough evidence to suggest that essential life processes such as metabolism, translation, splicing, etc. evolved around RNA. RNA used to act as a genetic material as well as a catalyst. There are some important biochemical reactions in living systems that are catalyzed by RNA catalysts and not by protein enzymes. But, RNA being a catalyst was reactive and hence unstable. Therefore, DNA has evolved from RNA with chemical modifications that make it more stable. DNA being double-stranded and having complementary strand further resists changes by evolving a process of repair. 6.4 Replication. While proposing the double helical structure for DNA, Watson and Crick had immediately proposed a scheme for replication of DNA. To quote their original statement, that is as follows: it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Watson and Crick, 1953. The scheme suggested that the two strands would separate and act as a template for the synthesis of new complementary strands. After the completion of replication, each DNA molecule would have one parental and one newly synthesized strand. This scheme was termed as semi-conservative DNA replication. Figure 6.6. 6.4.1 The experimental proof. It is now proven that DNA replicates semi-conservatively. It was shown first in Escherichia coli and subsequently in higher organisms, such as plants. Figure 6.6 Watson-Crick model for semi-conservative DNA replication 2022-23. 105 Molecular basis of inheritance. Figure 6.7 Meselson and Stahl's experiment in human cells. Matthew Meselson and Franklin Stahl performed the following experiment in 1958. They grew E. coli in a medium containing 1,5-NH4Cl. 15N is the heavy isotope of nitrogen as the only nitrogen source for many generations. The result was that 15N was incorporated into newly synthesized DNA, as well as other nitrogen-containing compounds. This heavy DNA molecule could be distinguished from the normal DNA by centrifugation in a cesium chloride CSCl density gradient. Please note that 15N is not a radioactive isotope, and it can be separated from 14N only based on densities E. Then they transferred the cells into a medium with normal 14NH4Cl and took samples at various definite time intervals as the cells multiplied, and extracted the DNA that remained as double-stranded helices. The various samples were separated independently on CSCl gradients to measure the densities of DNA. Figure 6.7. Can you recall what centrifugal force is, and think why a molecule with higher mass density would sediment faster? The results are shown in figure 6.7. Thus, the DNA that was extracted from the culture one generation after the transfer from 15N to 14N medium, that is after 20 minutes, E. coli divides in 20 minutes, had a hybrid or intermediate density. DNA extracted from the culture after another generation, that is after 40 minutes, 2 generation was 2022 minus 23. 106 biology composed of equal amounts of this hybrid DNA and of light DNA. If E. coli was allowed to grow for 80 minutes, then what would be the proportions of light and hybrid densities DNA molecule? Very similar experiments involving use of radioactive thymidine to detect distribution of newly synthesized DNA in the chromosomes was performed on Visia-faba beans by Taylor and colleagues in 1958. The experiments proved that the DNA in chromosomes also replicate semi-conservatively. 6.4.2 The machinery and the enzymes in living cells, such as E. coli, the process of replication requires a set of catalyst enzymes. The main enzyme is referred to as DNA-dependent DNA polymerase, since it uses a DNA template to catalyst the polymerization of deoxynucleotides. These enzymes are highly efficient enzymes as they have to catalyst polymerization of a large number of nucleotides in a very short time. E. coli that has only 4.6 times 106 pp compared with human whose diploid content is 6.6 times 109 pp completes the process of replication within 18 minutes. That means the average rate of polymerization has to be approximately 2000 pp per second. Not only do these polymerases have to be fast, but they also have to catalyst the reaction with high degree of accuracy. Any mistake during replication would result into mutations. Furthermore, energetically replication is a very expensive process. Deoxyribonucleoside triphosphate SERP.
दिया दीदी है फिर तो
106 biology composed of equal amounts of this hybrid DNA and light DNA. If E. coli was allowed to grow for 80 minutes, then what would be the proportions of light and hybrid densities DNA molecule? Very similar experiments involving use of radioactive thymidine to detect distribution of newly synthesized DNA in the chromosomes was performed on Visia-faba beans by Taylor and colleagues in 1958. The experiments proved that the DNA in chromosomes also replicates semi-conservatively. 6.4.2 The machinery and the enzymes in living cells, such as E. coli, the process of replication requires a set of catalyst enzymes. The main enzyme is referred to as DNA-dependent DNA polymerase, since it uses a DNA template to catalyze the polymerization of deoxynucleotides. These enzymes are highly efficient enzymes as they have to catalyze polymerization of a large number of nucleotides in a very short time. A. coli that has only 4.6 times 106 pp compared with human whose diploid content is 6.6 times 109 pp completes the process of replication within 18 minutes. That means the average rate of polymerization has to be approximately 2000 pp per second. Not only do these polymerases have to be fast, but they also have to catalyze the reaction with high degree of accuracy. Any mistake during
कृष्णा चुप हो जाओ
substrates, they provide energy for polymerization reaction. The two terminal phosphates in the deoxynucleoside triphosphates are high energy phosphates, same as in case of ATP. In addition to DNA-dependent DNA polymerases, many additional enzymes are required to complete the process of replication with high degree of accuracy. For long DNA molecules, since the two strands of DNA cannot be separated in its entire length due to very high energy requirement, the replication occurs within a small opening of the DNA helix, referred to as replication fork. The DNA-dependent DNA polymerases catalyze polymerization only in one direction, that is 5 feet 3. This creates some additional complications at the replicating fork. Consequently, on one strand the template with polarity 3 feet 5, the replication is continuous, while on the other the template with polarity 5 feet 3, it is discontinuous. The discontinuously synthesized fragments are later joined by the enzyme DNA ligase figure 6.8. The DNA polymerases on their own cannot initiate the process of replication. Also, the replication does not initiate randomly at 107 molecular bases of inheritance because of the requirement of the origin of replication that a piece of DNA if needed to be propagated during recombinant DNA procedures requires a vector. The vectors provide the origin of replication. Further, not every detail of replication is understood well. In eukaryotes, the replication of DNA takes place at S phase of the cell cycle. The replication of DNA in cell division cycle should be highly coordinated. A failure in cell division after DNA replication results in polyploidy, a chromosomal anomaly. You will learn the detailed nature of origin and the processes occurring at the site in high classes. 6.5 Transcription, the process of copying genetic information from one strand of DNA into RNA is termed as transcription. Here also, the principle of complementarity governs the process of transcription, except the adenosine complements now forms base pair with uracil instead of thymine. However, unlike in the process of replication, which once set in, the total DNA of an organism gets duplicated. In transcription, only a segment of DNA and only one of the strands is copied into RNA. This necessitates defining the boundaries that would demarcate the region in the strand of DNA that would be transcribed. Why both the strands are not copied during transcription has a simple answer. First, if both strands act as a template, they will code for RNA molecules with different sequences. Remember, complementarity does not mean identical, and in turn, if they code for proteins, the sequence of amino acids in the proteins would be different. Hence, one segment of the DNA would be coding for two different proteins, and this would complicate the genetic information transfer machinery. Second, the two RNA molecules if produced simultaneously would be complementary to each other, hence would form a double-stranded RNA. This would prevent RNA from being translated into protein and the exercise of transcription would become a futile one. 6.5.1 Transcription unit. The transcription unit in DNA is defined primarily by the three regions in the DNA, I, A promoter, E, the structural gene, E, a terminator figure 6.8 replicating for 2022-23. 108 Biology. There is a convention in defining the two strands of the DNA in the structural gene of a transcription unit. Since the two strands have opposite polarity and the DNA-dependent RNA polymerase also catalyze the polymerization in only one direction, that is, 5' pointing arrow 3, the strand that has the polarity 3' pointing arrow 5' acts as a template and is also referred to as template strand. The other strand which has the polarity 5' pointing arrow 3 and the sequence same as RNA except thymine in the place of uracil is displaced during transcription. Strangely, the strand which does not code for anything is referred to as coding strand. All the reference point while defining a transcription unit is made with coding strand. To explain the point, a hypothetical sequence from a transcription unit is represented below. 3. ATGC 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 minus 5' template strand 5. TACG 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 minus 3' coding strand. Can you now write the sequence of RNA transcribed from the above DNA? Figure 6.9 schematic structure of a transcription unit. The promoter and terminator flag the structural gene in the transcription unit. The promoter is said to be located towards 5' upstream of the structural gene. The reference is made with respect to the polarity of coding strand. It is a DNA sequence that provides binding site for RNA polymerase, and it is the presence of a promoter in the transcription unit that also defines the template and coding strands. By switching its position with terminator, the definition of coding and template strands could be reversed. The terminator is located towards 3' downstream of the coding strand, and it usually defines the end of the process of transcription. Figure 6.9. There are additional regulatory sequences that may be present further upstream or downstream to the promoter. Some of the properties of these sequences shall be discussed while dealing with regulation of gene expression. 6.5. Transcription unit in the gene. A gene is defined as the functional unit of inheritance. Though there is no ambiguity that the genes are located on the DNA, it is difficult to literally 2022-23. 109 molecular bases of inheritance define a gene in terms of DNA sequence. The DNA sequence coding for the now RNA molecule also define a gene. However, by defining a cistern as a segment of DNA coding for a polypeptide, the structural gene in the transcription unit could be said as monocystronic mostly in eukaryotes, or polysystonic mostly in bacteria or prokaryotes. In eukaryotes, the monocystronic structural genes have interrupted coding sequences. The genes in eukaryotes are split. The coding sequences or express sequences are defined as exons. Exons are said to be those sequences that appear in mature or processed RNA. The exons are interrupted by introns. Introns or intervening sequences do not appear in mature or processed RNA. The split gene arrangement further complicates the definition of a gene in terms of a DNA segment. Inheritance of a character is also affected by promoter and regulatory sequences of a structural gene. Hence, sometimes the regulatory sequences are loosely defined as regulatory genes, even though these sequences do not code for any RNA or protein. 6.5.3 Types of RNA in the process of transcription in bacteria. There are three major types of RNAs: mRNA, messenger RNA, and transfer RNA, and rRNA, ribosomal RNA. All three RNAs are needed to synthesize a protein in a cell. The mRNA provides the template, then brings amino acids and reads the genetic code, and rRNA plays structural and catalytic role during translation. There is a single DNA-dependent RNA polymerase that catalyzes transcription of all types of RNA in bacteria. RNA polymerase binds to promoter and initiates transcription initiation. It uses nucleoside triphosphate as substrate. Figure 6.10 Process of transcription in bacteria 2022-23. 110 Biology and polymerizes in a template-dependent fashion following the rule of complementarity. It somehow also facilitates opening of the helix and continues elongation. Only a short stretch of RNA remains bound to the enzyme. Once the polymerase reaches the terminator region, the nascent RNA falls off, so also the RNA polymerase. This results in termination of transcription. An intriguing question is that how the RNA polymerase is able to catalyze all the three steps, which are initiation, elongation, and termination. The RNA polymerase is only capable of catalyzing the process of elongation. It associates transiently with initiation factor sigma and termination factor rho to initiate and terminate the transcription, respectively. Association with these factors alter the specificity of the RNA polymerase to either initiate or terminate. Figure 6.10. In bacteria, since the mRNA does not require any processing to become active, and also since transcription and translation take place in the same compartment, there is no separation of cytosol and nucleus in bacteria. Many times the translation can begin much before the mRNA is fully transcribed. Consequently, the transcription and translation can be coupled in bacteria. In eukaryotes, there are two additional complexities. There are at least three RNA polymerases in the nucleus in addition to the RNA polymerase found in the organelles. There is a clear-cut division of labor. The RNA polymerase I transcribed the RNAs. Figure 6.11 Process of transcription in eukaryotes 2022-23. 111 molecular bases of inheritance 28S, 18S, and 5.8S. Whereas the RNA polymerase 3 is responsible for transcription of RNA, 5 is rRNA, and SNRNA small nuclear RNAs. The RNA polymerase 2 transcribes precursor of mRNA, the heterogeneous nuclear RNA, hnRNA. The second complexity is that the primary transcripts contain both the exons and the introns and are non-functional. Hence, it is subjected to a process called splicing where the introns are removed and exons are joined in a defined order. hnRNA undergoes additional processing called as capping and tailing. In capping, an unusual nucleotide methylguanosine triphosphate is added to the 5 end of hnRNA. In tailing, adenylate residues 200 to 300 are added at 3 and in a template-independent manner. It is the fully processed hnRNA, now called mRNA, that is transported out of the nucleus for translation. Figure 6.11. The significance of such complexities is now beginning to be understood. The split gene arrangements represent probably an ancient feature of the genome. The presence of introns is reminiscent of antiquity, and the process of splicing represents the dominance of RNA world. In recent times, the understanding of RNA and RNA-dependent processes in